This tutorial is going to walk through this practice problem about cell signaling. If you haven't looked it over recently, take a minute now to pause the video and read the problem, then come back to see how we're going to approach it. Since this problem focuses on protein kinase receptors, let's take a minute and review protein kinase receptor pathways. Many of these kinds of receptor pathways are activated by growth factors, so that's the example that we're going to use here. Before a signaling molecule or ligand arrives, we have an inactive receptor kinase and RAS effector protein, which is bound to GDP. Upon ligand binding, the growth factor receptors will dimerize and phosphorylate each other. This causes a change in the way that they interact with RAS, and this causes RAS to exchange its bound GDP for GTP, and RAS is thereby activated. This activated RAS will activate the first kinase in a cascade. As a reminder, a kinase is an enzyme that phosphorylates things, and so when you have a kinase cascade, it's a series of kinase enzymes that phosphorylate each other and can phosphorylate other target molecules. So in this case, the first kinase will activate a second kinase and possibly additional kinases downstream in the cascade, always by phosphorylating each other. The final kinase in the cascade will enter the nucleus and phosphorylate transcription factors that regulate gene expression. This leads to a change in the activity of these transcription factors and corresponding changes in gene expression. So now let's go back to the problem. Which of the following would affect the binding of a ligand to a protein kinase receptor? And in the first one, we're asking about the presence of a competitive antagonist. This one is pretty straightforward, because a competitive antagonist, by definition, binds to a receptor in place of a ligand and prevents activation of the receptor. So in this case, the competitive antagonist is itself binding to the receptor instead of the ligand. In the next part, we're looking at a mutation that prevents phosphorylation of the receptor. Phosphorylation is something that happens after ligand binding, and so in this case, it's not expected to affect the interaction between the receptor and the ligand, since that happens first. If we're looking at a mutation in an effector protein, again, effector proteins are things that are all activated downstream or after ligand binding to the receptor. So you could have any one of these effector proteins being affected by this mutation. All of these mutations are found in proteins that are activated after the receptor binds to the ligand. And so none of these mutations are likely to affect receptor-ligand interactions themselves. Finally, we have a mutation in the extracellular domain of the receptor. So that would be on the cell surface. Since protein kinase receptors are cell surface receptors, the ligand binding domain is on the extracellular side of the membrane so that it can interact with signaling molecules from other cells. So if you're making a mutation in the part of the protein that's in the extracellular region, then that's going to be the part of the protein that's responsible for binding to the ligand. And so a mutation in the extracellular domain would prevent ligand binding. As a reminder, these are the diagrams of the signaling pathways that you'll get as a reference for any questions on the final that relate to cell signaling. As you review how each one works, you can test yourself by asking these kinds of questions about each one. Either by yourself or with a study partner or group, think about what kinds of drugs you could use or what kinds of mutations you could make that would affect each signaling pathway, and try to predict their consequences. It's a great way to both test your understanding of the pathways and develop analytical skills.